Yeah, hello, good morning, and you're welcome back to the channel. We continue our discourse this morning. Now, we are going to spend a little bit of time discussing post mortem microbiology. Remember, we are discussing forensic microbiology, so we'll take our time to discuss post mortem microbiology. Now, what's post mortem microbiology? Now, this is the aspect of microbiology that puts its focus on looking at what happens after a person dies, the microbial communities within the human body. I had said earlier that when analysis wants to be done on a body of a person who has passed, samples can be taken from different parts of the body, from the mouth, from the gut, from the mouth, the gut, from skin. Swabs can be taken of the skin, even from inside the ears. So samples can be taken from different sides of the body. So when we say post-mortem microbiology, we are talking about the aspect of microbiology that wants to look at what happens to a body after it dies, what happens to the microbial communities that exist within a body after the body dies. Now, it's important, there's something called a post-mortem interval. Post-mortem interval. Very many times, forensic microbiology wants to work to be able to estimate the time of death of a person. This is important. It can also help, be complementary to the work which a pathologist will do, or somebody who does what do you call it, this analysis they do for people after they have died. This kind of analysis, they try to estimate the time of death of an individual. Now, in forensic microbiology, we could also help in estimating this time of death. And how could we help in estimating this time of death? We can use our knowledge of microbial succession, our knowledge of what happens to a body when the body dies, when the body passes away, what happens to the body. We can use that knowledge, and then when samples are taken from the body at the time when the forensic microbiologist comes in contact with the body, analyzing those samples can help the microbiologist to give a rough estimate, an estimate of when this person actually passed. Now, it's important to note that there are a lot of research facilities and sites, especially in the United States, that are focused on studying human bodies, dead bodies, cadavers. And the studies are looking at these bodies, that what happens when these bodies pass, when people pass, what happens to their body. And there's a focus for forensic microbiologists. Our focus is on the microbial communities. What happens to the microbial communities? Which organisms are more, which organisms are less? And then, over a period of time, what happens to the body. Now, it's important to note that the human body carries trillions of microorganisms, not thousands, not trillions of microorganisms. And these microorganisms are affected. When a person dies, a lot of things happen. Now, there is what we call autolysis and putrefaction. Putrefaction is your decay, normal decay. Now, when we say autolysis, this is a state where the enzymes within the body begin to destroy or lyse, break open the cells of the body. And this is because when a person passes, circulation stops, many things that were working before, the heart stops to pump blood around the body, and so the cells become starved. They become starved for oxygen, they become starved for glucose and sugars, and so these cells cannot continue to sustain life. And so before you know it, even the enzymes within the cells that were supposed to be catalyzing different reactions now begin to bring a breakdown of these cells. And this is what we refer to as autolysis or autolysis. And it occurs in a dead body. And then it leads and, flows on to putrefaction, which is decay or decay of the human body. And all of this is important. So, like I said before, post-mortem interval estimation is very important. We want to be able to estimate, if a forensic microbiologist comes in contact with the body, wants to be able to tell, when did this individual die? Was it 2 days ago? Was it 20 days ago? And the microbial community from the sites, from samples from the sites within the body that have been taken, would help to make these decisions, will help to give this information to the scientist. Now, there's a term which we call thanatoomicrobiome. Thanatoomicrobiome. This term refers to the microbial communities that exist within a dead body. Microbial communities that exist within a dead body, and we do a lot of analysis to be able to determine the kind of microbial communities that exist within a dead body. It's also important to realize that a succession occurs. That is, there is a gradual change in the composition of the microbial communities or that are within a dead body. And this gradual change, it can be measured, can be followed. Now, it's important that we realize that this change is not a random change. It's not a change that is left to chance. No, it is a change that follows a particular pattern. And it follows this pattern because this change, you know it follows the pattern. It follows a pattern because there are many factors, that affect the decomposition of a human body. The age of the individual involved, the state of health of the individual involved, all these things are factors. Age, state of health, temperature at which, then, where the body is the body out in the open, is it deep in the ground? All these factors would play a role in determining how a person is going to progress and what is going to happen with the death of a person. Now, it's important to realize that there are about four different stages of succession, microbial succession within a dead body. It takes about four different stages. I will run through these four different stages very quickly. The first stage is the stage of the initial stage. At the initial stage, that's between 0 to 2 days, microbial activity is really low. Microbial activity is really low, and the microorganisms have not started to work very much on the cells within the body within 0 to 2 days. So you don't have much smelling of the body, you don't have much bloating of the body. That's between 0 and 2 days. Now, what you have happening most of the time between that 0 and 2 days is that autolysis begins to occur. So the enzymes within the body and within the cells of the body begin to break down the different cells within the body. That's from 0 to 2 days. This is the initial stage. Now, after the initial stage, we have the putrefaction stage. The putrefaction stage is from day 2 to day 10. Now, the putrefaction stage, many microorganisms begin to act a lot on all the organic matter that are present in this dead body. Remember that the gut carries a lot of microorganisms. So the gut microbiome is carrying a lot of microorganisms, and from there a lot of microbial activity begins to happen. So a lot of activity is happening, and then because a lot of microorganisms are working, they are dividing, they are feeding on the organic matter, it begins to lead to many different circumstances. For an example, there's the release of a lot of H2S gas through the activity of these microorganisms. Now, a list of some of the microorganisms: your Clostridium, your Proteus, your Bifidobacterium, all these bacteria that we can find, yeah, even our bacteroidetes, all the bacteria that we can find in the guts, they begin to act, and then there's a buildup of H2S, that's hydrogen sulfide, and this is the point at which the body begins to swell. If you've not even seen a dead human body, you must have seen like the carcass of maybe a sheep or a dog or any animal that has been maybe hit by a car or something. You see that after a while the body begins to swell and there's a buildup of pressure, there's gas within the body, and this is due to the release of H2S. So you have the initial stage, 0 to 2 days. You have the putrefaction stage, which is from 2 days to 10 days. Then the third stage is the stage of active decay. So at this time now, a lot of body fluids are now flowing through the body, the cadaver or the carcass, because a lot of autolysis has occurred, and so many of the parts of the body have been broken down. Many of the organs have been broken down. The liver, the heart, they have gone through a lot of decaying. So you have active decay, where a lot of organisms, feed on all these organs, and you have a lot of facultative anaerobes really working at that time to feed on all these parts of the body. And this is the third section. Now, the fourth and final section is the stage of the active decay and skeletonization. So at this time now, decay continues, and that's after 20 days, and all the organic matter, whatever is remaining of the organic matter, the microorganisms continue to act on them, continue to work on them all the way till by the time 20 and 25 days is showing, the skeleton of that body now begins to show. Now, but like I said that all this depends on the state at which the body is. You understand? A body in a state of a bit of a warm temperature and a humid environment would decay much faster than a body that is in a very dry environment. So it depends on the place where the body is. First of all, that's going to play a role in how fast or slowly that body is going to decay. So all these parts of microbiology, that for which we, look at dead bodies, look at the succession, microbial succession within a dead body, is what we look at in forensic microbiology. And you might ask that what is the importance of this? Now, the importance of all of this is that many a times there could be some sort of litigation, there could be legal issues surrounding the death of an individual. In fact, an individual can be suspected to have been poisoned through the use of chemicals, through the use of biologic agents. So, if there is suspicion that an individual has been killed or has died under suspicious circumstances, all this kind of science, they play a role in trying to unravel the mystery of how this individual died. And so all of this is important, and that's what we do in post-mortem microbiology. Thank you for your time this morning, and do well to like this content, share this content, and let's build this community. Have a wonderful day.